Here we go. It's Friday. It's kind of cloudy, dreary Friday. So I'll try to make this as short and sweet as possible. This is the beginning of the end. This is the beginning of the last chapter that we're going to do in this course. And it's all about the endocrine system. So this is going to be an introduction to that system as well. And then we're going to get more specific over the course of, including today, the next 3 days. So our last Zoom meetings are going to be today, which is a Friday, Monday, and Tuesday in preparation for your final lecture quiz, which will be Wednesday. So let me call up my first board. And here we go. So I think it's chapter 16 in your book. I don't have my text at my side, so I'm just going to write the word endocrine system, or the term endocrine system. And we're going to just first build a mini glossary here. We're going to keep, so much of A&P is not rocket science in terms of concepts. It's learning a new language. So we're going to start to do that today. And I'm just going to write out exocrine. I'll expect you to know this. Exocrine versus endocrine. We went over this when we talked about glands way back at the beginning of the semester. Just to remind you, these describe usually cells or glands. Exocrine glands or cells usually secrete a fluid directly into a duct that is aimed at or directed toward its target, where it needs to go. Endocrine, which is what we're focused on in this chapter, is a gland or cells that secrete hormones, which are chemical messengers that are carried by the blood. So that's what we're doing. Now, just some other terms that you may read or hear me say. Autocrine, strictly speaking, the textbook definition of autocrine is a chemical that has its effect on the same cell that secretes it, hence the prefix auto. We also use this in an adjective form. So we might say an autocrine cell or an autocrine gland. That just means it's secreting something, synthesizing and secreting something that has an effect on the same cell that secreted it. This word, paracrine, refers to, strictly speaking, it's a chemical that has its effect on cells within its close proximity or neighboring cells. We also use this as an adjective. We might say a paracrine cell or a paracrine gland would secrete some sort of chemical that has its effect on cells right next to it or very close to it. So its secretions don't actually have to enter the blood and circulate throughout the entire body. I know I've defined this for you before. I'm just so we're all on the same page. What is a hormone? It's just a chemical messenger carried by the blood that must enter the bloodstream to do its traveling, to find its target site or target cell. There's two basic classifications of hormone types. And it's based on their chemistry, their biochemistry, how they are arranged chemically. The first one are referred to as amino acid based. Just like the name is describing, this class of hormones is made up of amino acids. So that's pretty straightforward and easy. We also call these, if you hear me say or you read peptide hormones, same thing, because a peptide is just a relatively short chain of amino acids. So we also call these peptide hormones. When I was in graduate school, we used the term, this isn't used so much anymore, but I might say it just because old habits are hard to break, protein hormones. And that's because proteins are made up of, guess what? Amino acids. So all of these things are sort of synonymous with each other. These are by far the most common class or type of hormones in the human body. The second class or type are steroids. We've all heard a lot about steroids in our lives. Once again, that's just referring to its chemical composition, what it's made up of. So steroids are made from cholesterol. That's their chemical composition, and they look like cholesterol rings, steroid hormones. Cholesterol itself is lipid-based. We learned that when we were learning the 4 organic compounds in the human body. So basically, we have amino acid-based hormones and steroids. They behave differently. Those are the 2 main classes of hormones in the human body. Another word, I'm just going to go call up a new whiteboard here. Another term. It's hard to write up at the top of this board with this plastic pen. That's where my writing gets the worst. It's leukotrienes, L-E-U. Leukotrienes are a class of chemicals. They're chemical messengers. They're strictly not considered hormones. These are they're also lipid-based. I may have mentioned these when we were talking about lipids the first week of class. They're typically secreted oftentimes by white blood cells, but their function is they mediate the inflammatory process in the human body. So I'm just going to write they mediates inflammation. Usually when we have some sort of damaged tissue. We'll talk a lot about leukotrienes. They're sort of a special class of chemical messenger. All right, let's go on to hormone actions at the target cell. So a hormone is secreted by a gland, a group of cells. It has to enter the blood, travels wherever blood goes in the human body. So a hormone basically gets secreted, dumped into the bloodstream, and then it goes everywhere that our blood goes. And then it sort of finds or looks for its target cell. And it usually does some sort of binding at that target cell. What this means, hormone actions at the target cell. I'm just allowing another student to drop in here. What that means is hormone action at its target cell. What happens at the target cell when a hormone binds to it or within it? That's what we're talking about. So there's usually an alteration. So they alter the activity of or at the target cell. And they usually do this. There's examples, there's 4 main examples. I'll write them 1, 2, 3, 4. They can alter the permeability of the target cell membrane. So change permeability. I'm just going to write of T cell. No, I'm not going to write T cell because then you'll get confused. You might think I'm talking about the immune system. Target cell membrane. I'm always looking for ways to get through this more quickly because there's so much information. Second, they might stimulate enzymes within the target cell. They might turn on enzymes, sort of enzyme synthesis or activity. A third possibility or very common action is they might induce the target cell to secrete something. So I'm just writing induce secretions. And finally, they might initiate or stimulate mitosis. Something else they might do is, I'll give you a fifth one, stimulate protein synthesis. Which is similar to number 2, but it's worth listing as its own separate activity. So next, it's going to sound the same. This next topic is going to sound the same as what we just titled this, hormone actions at the target cell. But the next thing I want to say is what a hormone does with its receptor at a target cell, how a hormone binds to a target cell receptor, and then what happens. So I'm going to write hormones and target cell receptors and activity. So now we're not just discussing what the overall target cell might do once a hormone gets to it, binds to it, but we're going to be more specific and look at what type of binding and activity actually occurs within the context of this. Within the, sorry, I was trying to get back to this board, within the context of these type classes of hormones. All right, so losing my way here. There we go. There's just 2 ways that hormones really behave at their receptors at the target cell, and they're completely based on whether or not they're amino acid-based hormones or if they're steroid-based hormones. So I'm just going to write in number 1 and list this as water soluble hormones. These are the same thing as that class of amino acid-based hormones. Amino acid-based hormones, peptide hormones, protein hormones, are water soluble. That's what causes them to bind the way they do at the target cell. The easiest way for me to explain this, I'll write a few things and then I'm going to draw a picture. So first, they act on a receptor on the plasma membrane of the target cell. You can read all of the specifics about this in your textbook. Now I'm going to draw a picture for you because it's much more beneficial, in my opinion, for a student to learn this process if they can just watch it happen. So this is going to be our target cell. It can be any cell in the human body. Hormones are very small, they're tiny. They're chemical messengers. They're not as big as a cell itself. So I'm just going to make a hormone out here. And let's make it a happy hormone because it's Friday. And let's make it an amino acid water-based hormone. That's important. The plasma membrane of our cells are phospholipid bilayers. That's a Bio 1 topic. So there's that layer, that bilayer of lipids squished between 2 phosphate layers. Point being is that water-soluble things cannot just easily enter our cells. Water-based things cannot just enter target cells. So in this case, an amino acid water-based hormone cannot enter the target cell itself. It must bind to a receptor out here specifically for that hormone. So this hormone is traveling through the blood. It finds this target cell, which has a receptor on its surface specifically made for that hormone. It binds to that receptor. Because it can't enter the cell directly, it has to bind to the surface receptor. I know I keep repeating myself. This then launches what we call a second messenger system. I think I mentioned that in the nervous system a long time ago. Now we're going to look at this from a hormone-based context. This receptor, this surface receptor, is typically coupled with a G protein. You've heard this, a little bit of this story in the past. So on the inside of the cell, the interior of the cell, this G protein is physically coupled with the receptor. And when the hormone binds to the receptor, it activates this G protein. Usually in G protein activation, what happens is G protein is inactive when GDP is bound to it, the diphosphate. And when this hormone binds to the receptor, it activates this G protein when GTP is bound to it. Nevertheless, this becomes activated and it sets off a cascade of events on the interior of the cell. G protein is coupled with something called adenylate cyclase. This is an enzyme that gets activated through the activation of G protein. And then when activated, adenylate cyclase enzyme, it's just a cascade of reactions. We're just tipping over the first domino here, and then it tips over this domino, and then this domino. That's why we call it a cascading reaction. When activated, adenylate cyclase converts ATP to cyclic AMP. So I'm just going to write little c. Now, AMP, you know what ATP stands for, adenosine triphosphate. AMP just stands for adenosine monophosphate. So essentially, when we convert ATP to cyclic AMP, we're cleaving two phosphates off of that ATP molecule. It gets its name cyclic because of its shape, sort of circular shape. This is the second messenger in this second messenger system. So we've just set off a domino reaction here to activate this second messenger. The first messenger is the hormone out here. So the reason we call the second messenger system, there's the first one. The second one is the activation of cyclic AMP. And the reason is because once that is activated, the second messenger, it typically activates inside of the cell. It activates what we call kinases, which just basically means cellular chemicals that alter the metabolism of the cell. What they do is they typically phosphorylate. It phosphorylates proteins, which alters the metabolic activity of this cell, either increases the metabolism of this cell, or it can also inhibit it. So last thing I'm writing here is it alters cellular metabolism. I know this is a long story, but it just goes step 1, binding to a surface protein because it can't enter. The G protein coupled with the surface protein activates adenylate cyclase, which is an enzyme that converts ATP to cyclic AMP, which is our second messenger now. Cyclic AMP activates this class of cellular chemicals, which typically phosphorylates proteins and alters cellular metabolism. Oftentimes it increases the metabolism of the cell. What does that mean, increasing metabolism? Say, for example, it might ramp up the production, the overall production of this cell to perform cellular respiration and produce more ATP. This is a very typical second messenger system, but it's not the only one in the human body. So I'm just going to mention we just saw what a G protein can do. Or a G protein, rather than activating adenylate cyclase and cyclic AMP being the second messenger system, a G protein may also release calcium stores. I'll just say can also release calcium ions from storage inside of the cell, which can activate calmodulin. We talked about that protein when we discussed smooth muscle cells. And then calmodulin is a calcium binding messenger protein, which then activates enzymes. So there's other second messenger systems. So calcium can also be a second messenger. Cyclic AMP is not the only one. So the cell typically releases second messengers in response to extracellular signaling molecules, first messengers like hormones. All right, that's going to take care of just moving back. I should go back two boards here. Number 1, that's how water-soluble hormones behave. They're amino acid based. They act on a receptor on the plasma membrane of the target cell. Why? Because they are water-based, they're water-soluble, and they can't enter the cell. The second hormone and target cell receptor activity are steroid-based hormones. So we're just looking at those two classes, amino acid-based and how they behave with the second messenger system, and now steroid-based are going to behave differently. So let's explain that. So this is steroid-based activity of the hormone when it binds at the target cell. So let's draw a new target cell. Here's our steroid hormone out here. Let's make it a different shape. Triangular shape. Now, this lipid-based, cholesterol-based hormone still has to travel throughout the bloodstream in order to find its target site. But it is lipid-based, not water-soluble, but lipid-soluble. And because this plasma membrane of our cells has that lipid bilayer, things that are lipid-based or lipid-soluble, guess what? They can slide right through this membrane. So they can enter the cell directly. So now this steroid is inside of the target cell because it can just slide right through that plasma membrane. I'm going to draw a nucleus because we'll end up talking about that in a minute. Sorry about the writing. I know it's frustrating for me too. So there's our steroid-based hormone. Once it enters the cell, it can bind to a receptor in the cytoplasm, like this. There's a receptor specifically made for that hormone, or it can, guess what? It can enter the nucleus and bind to a receptor made for it in the nucleus, like this. So this is our receptor, and we call, when the steroid or the hormone binds to its receptor, we just call it the hormone receptor complex. It doesn't matter per se if the binding occurs in the cytoplasm out here or once it gets into the nucleus in here. Either way, the hormone receptor complex typically, this is not the only thing a steroid does, but it's very common, which is why most textbooks use it as an example. So now we have a hormone receptor complex that enters or is in the nucleus. Like I said, it doesn't matter if it binds to the receptor out here and then goes in, or if it goes in first and binds to a receptor in here, okay? So we've got a hormone, steroid hormone receptor complex that's now in the nucleus. What's in the nucleus of a cell? DNA, or genetic information. So I'm going to draw a strand of DNA, chromosome, there it is, the double helix. And way back, the very first couple days of class, we talked about protein synthesis, transcription, and translation. Remember, when a protein needs to be synthesized, first we make a copy of, I'm just going to write the word transcription. We make a copy of that gene into an single-stranded RNA form. That's called transcription. And then that messenger RNA, after it's processed, makes its way to a ribosome in the cytoplasm where translation occurs. And we make a protein. We went through all of these steps from transcription, through translation at the ribosome, to linking those amino acids together to form a primary structure of the protein. Guess what? A lot of steroid hormones in the human body are the chemical signaling impetus initiators of protein synthesis. So I'm going to back up to my story here. So now we have our steroid hormone receptor complex in the nucleus. It typically seeks out a specific section or gene of our DNA to initiate this, the unraveling of the DNA. It gives the green light to this whole process of protein synthesis. This is the initiation point out here. So that's the typical behavior of steroid hormones. They usually diffuse right through the target cell membrane because they're lipid soluble. They bind to intracellular receptors, either in the cytoplasm or in the nucleus. And then what we say is they typically turn on a gene for protein synthesis. Now, what most of us hear about when we hear the word steroid, you think of people who take steroids, like bodybuilders or athletes who take steroids in order to enhance their strength, their muscle mass, whatever it is. Guess what? Those are called anabolic steroids. Anabolic means to build. And what we're doing here is we're making proteins and we're building mass. We're building muscle mass. Now that's just one small example of what protein synthesis does. It might do, our cells all over the human body are just constantly cranking out proteins all day long. So hormones are one of the typical initiators of that protein synthesis process. Okay, now I need to start a whole new topic, enhance our vocabulary, and try to plow through some more of this stuff for the day. So I'm going to call up a whole new set of whiteboards and start building more of a glossary term. So this term is upregulation. What does that mean? This is an example of when low levels of a hormone act on its target cell and cause its target cell to form additional receptors for that hormone. So if we have a target cell, we have a small hormone, when this hormone finds its target cell and binds to its receptor, the act of it binding causes the target cell to put more receptors out on its surface after that first binding. This is called upregulation, the enhancing of the effect of the hormone by the target cell producing more receptors for that hormone. That is opposed to downregulation, which you can guess almost has the opposite effect. Downregulation is just defined as when a target cell is under prolonged exposure from a hormone. So here's a hormone, it binds to, let's say there's obviously more than one binding to receptors on its target cell. So prolonged exposure of this process to the target cell sort of causes the target cell to reduce its numbers of receptors in order to downregulate the hormone's effects. It's sort of like sensory adaptation. The more this target cell gets bombarded with hormone, the less of the activity that hormone is going to have, and it's due to the elimination of, sorry, I didn't mean to erase the whole cell. It's due to the elimination of receptors for that hormone on the target cell itself. So there's sort of opposite effects. This one is an enhancing effect, and this one sort of downregulates or limits the effect, minimizes the effect of the hormone at the target cell. Next, I'm writing 3 types of endocrine gland stimuli. What does all of this mean? I'm trying to write the same headers that are in your book. And when you just read this, it would be helpful if you could make some sort of connection as to what this really means. What we're talking about now is what happens at the endocrine gland that synthesizes the hormone. So what this section in your book is what causes, what stimulates an endocrine gland to synthesize and secrete a hormone. So we're going to the opposite end of the chain of system from the target cell. What causes an endocrine gland in the first place to synthesize and secrete a hormone? And there's 3 basic main types of stimuli that can be put onto an endocrine gland to actually start secreting a hormone in the first place. The first one we call humoral stimuli. I believe we've mentioned this word in the past. Humor just means not funny humor, haha, but it means fluid. it's a really antiquated, old-fashioned word. Before we knew much about the human body, and I'm talking about going all the way back to like Greek and Roman times, the word humor referred to the fluids in the human body. So humoral stimuli really, in this case, just means from blood, which is one of the body fluids, from blood levels, say, for example, of certain ions in the blood. Could be a nutrient. Classic example, and since we've already done the skeletal system, is calcium levels in the blood. If you remember, when calcium blood levels drop, it stimulates parathyroid hormone to be secreted from the parathyroid gland. So calcium levels, when they decrease, when they drop, it stimulates, I'm just going to write PTH, parathyroid hormone, to be secreted. And that eventually raises blood calcium levels. Another example would be insulin acting on pancreatic cells. So when insulin levels drop, it stimulates pancreas cells to secrete the hormone insulin. So I'm just going to write insulin levels also in the blood would stimulate pancreatic cells, beta cells in the to secrete insulin. So those are examples of humoral stimuli coming from the blood. Another form of stimuli, I'm just going to overlay this, is neural stimuli. So a neuron or a group of neurons can innervate endocrine glands to secrete hormones. So I'm just going to write nerve fibers, stimulate hormone secretion and synthesis and secretion. And I'll give you some examples of this. Classic example is, we just learned this, and you're going to be tested on this topic today. So stress causes, I'm just going to write ANS, the autonomic nervous system, to innervate or stimulate the adrenal medulla. Remember, this is the sympathetic division of the nervous system. So stress causes the sympathetic nervous system to stimulate the adrenal medulla to release the hormones epinephrine and norepinephrine. Remember, norepi can be a neurotransmitter or a hormone. And you know what these things do. That's the sympathetic nervous system reaction. Dilates bronchioles, dilates pupils, increases blood flow to the heart and skeletal muscles, increases heart rate. All that long list of things that the sympathetic nervous system does. So the third type of stimuli on an endocrine gland, causing it or signaling it to synthesize and release hormones, is hormonal stimuli itself. All we're saying is that one hormone may travel to its target cell, which might be another endocrine gland, and stimulate that second endocrine gland to secrete another hormone. So it's just hormones stimulating release of other hormones. we call these first hormones tropic, spelled tropic, but we pronounce tropic. So tropic hormones are hormones that when secreted, they cause the release of other hormones somewhere else in the body. We're going to go through this whole system. The classic example is hormones from the hypothalamus. Just going to give you the hypothalamus might secrete releasing hormones that act on the pituitary gland. They release hormones that might go to, say, for example, the gonads in the human body, the ovaries or the testes, and stimulate those to release steroid hormones called estrogens and progesterones. So here in this chain, hormones from the hypothalamus acting on the pituitary, they would be tropic. And the hormones released from the pituitary and acting on the gonads would also be considered tropic, because in both cases, they're causing the release of other hormones from that target site. Okay, some more terms. We're building our glossary some more. That's supposed to read permissiveness. P-E-R-M-I-S-S. I-V-E-N-E-S-S. This word, permissiveness, isn't what you think it means. In this case, it's a hormone that cannot exert its full effect on its target cell without the combined action of another hormone. So here's our target cell. Here's our hormone. Let's say it binds to its target receptor on that target cell, and it causes a minimal effect. But if this hormone, let's just say out here, make it a different shape, also binds to the target cell, it enhances the overall effect of this first hormone. That's called permissiveness. It's a combined action of two hormones to enhance an overall effect. Classic example, these definitions, by the way, are in your book. Classic example are reproductive hormones and thyroid hormone. Both, these are two different reproductive hormones. We mean steroid hormones coming from the gonads, estrogens and testosterone. They typically work on cells in conjunction with thyroid hormone, it's hard to say quickly, in order to enhance the overall effects at that target cell. That's different from something that we call synergism. Synergism is a term that when more than one hormone produces the exact same effect. It doesn't enhance the effect of one hormone or the other. More than one hormone produces the exact same effect at the target cell. Classic example of this might be something like both, so I'm going to write example, both glucagon, which is a hormone that's secreted by the pancreas, and I'll talk about that later, and epinephrine, Both of these act on the liver and have the exact same effect. Causes the liver to metabolize glycogen, stored glucose, and release glucose into the blood. So there's a classic example of synergism. And finally, I'm going to try to save space here. You might guess what this means. Antagonism just means when two hormones have the opposite effect at the target cell. Or just two hormones have opposite overall effects. Classic example of antagonistic hormones are glucagon and insulin. They're both secreted by the pancreas. Glucagon, its overall effect is it increases blood sugar levels, blood glucose levels. Insulin has the opposite effect. It decreases blood glucose levels. I'll talk about how that works later when we're just describing hormones, endocrine glands, the hormones they secrete, and their activities. The last thing I'm going to do in the last 5 minutes is begin to talk to you about the hypothalamus-pituitary relationship. I probably won't get through all of this. You know, both of these structures are located in the brain, the human brain. Hypothalamus lies right smack, almost dead center in the inferior portion of the diencephalon. So I'm just going to draw a really big hypothalamus. Sort of has this weird W shape to it when we look at it in cross section. And then the pituitary gland hangs down from the hypothalamus, sort of a bean-shaped structure. So this is pituitary, hypothalamus. Like I said, I probably won't be able to do this whole lecture today. This is the major neuroendocrine gland in the human body that controls homeostasis. It also controls what happens here at the pituitary gland. This stalk of tissue, I need to erase this little piece because it's not going to sit well with how I want to describe this relationship. This stalk of tissue that the pituitary hangs from the hypothalamus is called the infundibulum. And the pituitary gland scientifically is known as the adenohypophysis. The hypothalamus is scientifically called the neurohypophysis. See if I can write legibly up here. All of this tissue in the hypothalamus is we call it neuroendocrine tissue, and we call this a neuroendocrine gland because it has neural tissue and hormone secreting tissue all in the same organ. Now the pituitary, the adenohypophysis, is divided into two sections like this. If this is the front, if I'm facing this way and you're looking at my profile, and you're examining my brain, this would be my face or anterior side of my head. This is posterior. So we call this the anterior pituitary, And this neurohypophysis is the same tissue as the hypothalamus, so we call this the neurohypophysis. Sorry. Down here, posterior pituitary. We usually just use these terms adeno and neurohypophysis when we talk about the pituitary. The reason I wrote this up here is because guess what? They are all the same tissue. And in order to understand how this relates, I'm going to draw this all in blue. So the posterior pituitary is the same exact tissue as the hypothalamic tissue up here. Now I'm going to erase neurohypophysis from here. And when I call the posterior pituitary the neurohypophysis, now you know why. It's because it's the same tissue that we have in the hypothalamus. All right, so that's step one. These, the anterior pituitary, the adenohypophysis, I'm going to use for red, and the neurohypophysis, the posterior pituitary, let me write the word anterior pituitary, and I'll draw this in red. They are basically separate structures. Even though we call the pituitary gland one structure, it's really two. And the posterior pituitary, the neurohypophysis, is really more part of the hypothalamus than it is part of the anterior pituitary. That's going to be key. These anatomical specifics are going to be key in understanding how they function. So I'm going to cut it there for the day. I'll draw this picture again tomorrow very quickly, meaning the hypothalamus and how it relates with the infundibulum and the anterior and posterior pituitary. And I'll start explaining how this organ or structure communicates with this structure down here and what the results are. So I'm going to stop my recording now.